fungi. So fungi are incredibly diverse, very, very widespread group of organisms. They have their own kingdom. You have the plant kingdom, the animal kingdom. There is a fungal kingdom. Well over 200,000 known species of fungi. Only about 8,000, luckily, that are known to be pathogenic or capable of causing disease. Most all fungi are what we call saprophytes. A saprophyte means it eats dead stuff. It doesn't eat living stuff. It doesn't get inside of a host and cause disease. These are our decomposers, and they help make the world go round. Without these types of fungi and some of the bacteria and even some things like worms and some of the insects, we would not have dead material decomposing, breaking back down into the essential elements to be reused in the environment. So these are essential for helping plants and animals to survive down the road. We have a lot of different fungi, most of which are microscopic that you'd probably never even know. You might recognize if you start to sneeze a little bit here or there. Maybe some of them are triggering some allergies. But for the most part, we'd never see any of them. The ones you are probably most familiar with are the mushrooms, the rusts, the conchs that you might see growing off the side of a tree, or bracket fungi. Those are all reproductive structures of just one type of fungi. There are many more out there that you would never notice, and the great majority do no harm, are actually very, very beneficial. But then there are some that can certainly cause plant disease, some serious diseases. They do have very weird bodies. You can see up there in the corner the body of a fungus. That body is called a mycelium. So there's no head, there's no arms or anything like that, but you have a body that can excrete enzymes into its environment, decompose or break down its food source, and then absorb it and use it internally for nutrients. This is very similar to what we do. The only real difference between fungal cells and our cells, which are incredibly similar, is that we do our digestion inside of our bodies. They digest outside of their bodies and then absorb it into. The long, thin strands of the body, so long, thin, multicellular strands, are called hyphae. Usually you'd never even see one hyphal strand, but thousands and thousands of them all jumbled up on top of each other, you can see in many cases forming a mycelial body. And this is sometimes what we're looking for on plant parts that have symptoms of disease. We're looking for signs if we suspect a fungal pathogen, and those signs would be then body parts. Some of those body parts might be mushrooms. Not all are associated with disease plants, but some could be. But definitely looking for mycelium or the hyphae associated in the same spot as a disease symptom gives us a clue. Signs, symptoms put together, maybe that's causing the problem in that particular plant. There are so many different types of fungal diseases out there that we cannot begin to go through every single one of them. It would be hard to do that. For the most part, what master gardeners need to be able to do is take a look at a plant sample and determine if they think that there could be a plant disease, number one. And number two, do they think it's bacterial, viral, or fungal? You don't typically have to speciate in most cases to be able to help, say, a homeowner. But some things to keep in mind when it comes to fungal diseases before we talk a little bit about symptoms is that they spread by wind, by water. They can spread their bodies around, and they can even produce, in some cases, structures that help them over winter, maybe even over summer, at least deal without having a host, and those are called sclerotia. Best way to put it, they look like rat droppings. Not all fungi produce them, but they are very hardy, surviving bad environmental conditions or without a host until conditions are right, and then that will start to sporulate, produce the mycelium, the body of the fungus, and it can restart its life cycle. Some of the common places we see this is in cabbages, let's say. You might see some of those sclerotia laying around. But not all fungal diseases will produce that, and most will not. But those would be signs if you were to see those. Looking under a microscope, if you were to see spores of fungi, those would be signs. So we're able to see visual a manifestation of the body of a pathogen compared to symptoms. We do have some weird things out there over in the protists. Some of them act like plants, some of them act like animals, and some of them act like fungi, but just are not related enough to be placed into one of those other kingdoms, so we kick them over into the protists. But we study them along with fungi because they do look and act so much like them, and some can cause plant diseases like downy mildews, pythium, which you may find in seedlings, with damping off diseases, turf grass like you see down there in the bottom, phytophthora, which causes a lot of issues with the roots of plants, anything from citrus all the way over to our veggies, and then club roots, which you might find in things like cabbage. So symptoms. Initially, early on, the symptoms of fungal disease can remind you of drought or even severe starvation where the plant has not been fertilized enough. The plants start to kind of appear off color, don't really look like themselves. They are weakened, very much susceptible to attack at this point in time. You'll start to see wilting, dieback from the tips, like some of the pictures that you see here on the side of the slide. We tend to talk about foci or focal points. In that big crop up there at the top, you see a huge focal point where you had a lot of diseased plants. But we can talk about individual foci or focal points just like in that middle picture there. If you noticed, you see very obvious the wilting and the dieback where you have necrosis or death of the tissue. There's symptoms there. But then look a little bit closer on that one prominent leaf there toward the bottom of that picture. You see the white area down next to the base of that leaf? That's fungal mycelium. That's a sign. So we've got signs associated directly with symptoms of fungal disease. And that's a focal point that we can talk about. Again, usually with fungal pathogens, the younger plants are usually killed very quickly, tend to be more susceptible than older plants. Older plants that get fungal pathogens, usually they decline over months, if not years. It can be kind of a slow death. If we're talking about impacts on roots, we tend to see some brown streaks, whereas roots in most cases should be white or cream color, maybe even a little bit yellowish, but streaks, brownish streaks running through those roots tells us we have some sort of malfunction, very oftentimes associated with a fungal infection. So again, here you can see just some of the foci, whether that's localized to a leaf or to an entire field of plants, and some of the symptoms that can be associated then with fungal plant pathogens. Bacteria. So bacteria are microscopic. You're not going to see them with the naked eye. They do have some hallmarks. They can look a little bit similar early on to some of the fungal infections, but there are some tips and tricks to help you set these aside from each other. They really, in most cases, like warm temperatures and very moist conditions. So, you know, if temperatures get kind of high and we've got a lot of moisture, bacteria as well as some fungal infections can be more problematic at those times than others. They're capable of reproducing themselves very quickly. They can split themselves to make babies. So they're cloning themselves, so you can get a exorbitant amount of bacterial cells very, very quickly. So we tend to see these diseases progress quite rapidly in most cases. You see the beaker of water up there in the top, and you see a stem. If you had something like pepper plant or tomato plant that comes in to the plant clinic and you suspected that had a bacterial disease, based on its symptoms. Are you going to see its signs? You're not going to see individual cells. These are microscopic. But if I were to cut that stem, put it into that beaker of water, and I wait just for a few minutes to see if I get that ooze, that streaming that you see running down through the middle of that beaker, that's a copious amount of millions of bacterial cells all jumbled up on top of each other, so many that they form a mass that I can see visually. Those are signs associated with the symptoms of a wilt in that particular plant. It's pretty unusual to be able to see bacteria in any other way, but this is one of the most definite ways. And yeah, would you kill the plant in that case to see this potential sign? Absolutely, but you got to sacrifice some to save the rest in some cases. So bacterial diseases, they tend to be less common than fungal or viral diseases. Bacteria, again, are kind of like fungi. They can be parasites, meaning they live off of another host, and then they could also be a pathogen, capable of causing disease. A lot of them are saprobes, they're just decomposers. There are even a handful of them that are autotrophs. Just like plants, they have chlorophyll, which means they're capable of doing photosynthesis, and they're beneficial in most all cases. Symptoms of bacterial disease: cankers, which look like sores. Just think of a canker sore. So sores, wilts, a very quick wilt, shoot blights, spots, individual little spots that are not associated with fungal signs, scabs, where it almost looks like a little sore spot with a scab, even could be a little bit corky if you were to touch that area, kind of spongy, soft rots, and galls. Those are all very common symptoms of bacterial infection. Again, these need wounds or natural openings to infect the plants, so they're usually very limited in where you find them on the plant. They have trouble moving across the veins, let's say, and you end up with these angular lesions like you see in the bottom picture, the leaf with the red arrow. That's a very angular lesion showing you necrosis right in the middle of the chlorotic area. That's where the actual infection has taken place, the colonization, more than likely reproduction of the pathogen, and so you've had some cell death in that particular area. That may or may not spread. That one spot might not be an issue to that entire plant. If it's capable of spreading over into the vascular tissue, it can certainly become systematic and run throughout the whole plant, causing disease. So, control methods. When it comes to control, we're usually talking cultural control. So what could we do to remove some aspect of that plant disease triangle when we're talking bacterial diseases? Because we do know antibiotics can be used in some cases to treat diseases, and sometimes in plants, that's only going to be done on a very large scale. For some, not all, but some very serious situations in agronomic crops. So we don't see that on a large scale in plants. So, we do then, over here, I just want to kind of show you the difference between some signs and symptoms of fungal and bacterial rots. So, you see here these two pictures, one of the bacterial soft rot, one of a white rot. So that bacterial soft rot, just as the name implies, that head of cabbage is starting to fall apart. It's mushy. It's soft, and I can promise you it has a very pungent odor to it, a rotten smell. If you were to compare the way that looks, at first glance it looked very similar. But you compare that with the white rot, you see necrotic tissue there, but you also see that white substance. That white substance is fungal bodies. That's signs and symptoms, the rot. On the bacterial side of things, you see symptoms, but are you going to see signs? No, not in this particular case. But this would help you to start to differentiate between whether or not you had a fungal or bacterial infection causing this particular disease. Viruses. I absolutely love researching and talking about viruses. If I had to do it over, I'd probably become a virologist. Viruses are submicroscopic. Note I say entities in this slide. I do not say organisms. Organisms implies that it's a living thing. Viruses, and this is arguable among some scientists, but viruses are non-living entities. There is no cell. There is no cell machinery. They don't look or behave like bacteria or viruses or even your cells, but they can manipulate living cells, and this is how they can cause disease. They can infect an individual host plant and become an obligate parasite, meaning they must have that host cell to perpetuate themselves. Notice I don't say reproduce. Cells have machinery to reproduce. If a virus has no cell parts, how can it reproduce itself? It cannot. So, think about it this way. Most everybody listening out there has gotten a viral infection, probably many throughout your life. Was that caused by one viral particle? No. That was caused probably by millions, if not billions, of viral particles moving throughout your body, affecting certain tissues, causing symptoms of disease in your body at that point in time. Okay? How were those viruses replicating? Not reproducing, but literally being copied, almost like a Xerox machine, copying exact copies. They are the ultimate terrorist because they hijack the cell's machinery once inside, and no longer can that cell take care of itself or make more of itself. It now makes the viral particles and replicates the viral particles. Once that cell then fills with viral particles that it's made, it bursts open, it dies, viral particles pour out into water, they pour out potentially into blood if we're talking animals, and are capable of then infecting other host cells. One of the first things we see in virally infected plants is the production of chlorophyll starts to cease. So the very earliest symptoms of viral infection in plants is chlorosis at the point of infection. And again, this can happen through wounds. So this could also, include places where we've pruned, where there's been wind abrasion, where you've had insects feeding, where you clipped off a leaf or took a rose off of your rose plant to put into your holiday decorations in some cases. Anywhere there's been a wound or a natural opening, you can then get a viral infection, which could then spread from one cell making or replicating the viral particles to another and another and another. Later down the road, then we tend to get necrosis or death in that particular area. Same thing happens in animals. If you think of herpes, which is the gift that keeps on giving, if you were to get a canker sore on your lip, that area is sore because viral particles are being replicated by the cells in your lip in that particular location, and they die as a result. Remember, they can't take care of themselves anymore or make more themselves. They're replicating the virus. Then they die. And the virus then is spread to other host cells in that particular area, and the process continues. So you get a sore in that area, or in the case of plants, the earliest symptom would be chlorosis. Same type of concept. Now, a lot of times what we see with plant viruses then, the cells are either going to grow or divide very rapidly, or maybe slower than they should be, will be unable to divide. So we see things like stunting beyond the chlorosis. The fruit may be stunted, the plant may be stunted, the roots may be stunted in some way. We see stunting. We see deformations in the fruit, in the leaves, in the stems. We see the plant twisting and distorting and doing very weird things because there are cells in very particular places being affected and killed. And that causes then the morphology of the plant and the fruit to be changed. So again, a malfunction can cause yield losses, and appearances can change. Not all viral plant pathogens are fatal. Some we've learned to tolerate. If it doesn't kill the plant or cause something undesirable, sometimes we keep it around. It's really hard to culture plant tissue and very costly to get viral particles out of it that we don't really consider causing a problem. If you like garlic, like most of us do, every bit of garlic that you consume is infected with a plant virus. That virus is host specific, like all of them. It can't hurt you. It's not even really causing anything that we know to be a problem in the plant, so we don't worry about getting rid of it or trying to prevent it. There are some tulips that have color break in them. Not all are caused by viruses, but some are, and we go, hey, we like it. We like this color break, or we like plant color variation, or we like a smaller plant, or we like to have a gourd or a pumpkin with these weird nodules on it. When it gets close to Thanksgiving, it looks cool on our table, or weird color breaks in those particular fruits. We like it. Doesn't kill the plant. We keep it around in some cases. So here are some electron micrographs. So we're talking magnified well over 250,000 times to see viral particles. You can see up in the top corner something that looks like an alien. That's called a bacteriophage. bacteriaphage. A bacteriophage is a virus that can only attack and infect and kill bacteria. Not us and not plants. So some of these are being bioengineered to try to help us control bacterial diseases. There are some entities smaller than viruses, like prions. Prions are not known to cause any sort of plant diseases, but we do know they cause some animal diseases, like Creutzfeldt-Jakob disease, mad cow disease. There's a variety of others. That's just a misshapen protein. Again, that's not a thing that's alive, but it's capable of causing disease because it has an impact on the cell. A virus is just a piece of DNA wrapped in a protein coat. That's all that it is. There's no other cellular components whatsoever to a virus. So it's a piece of DNA or RNA, a molecule of heredity, wrapped in protein to protect it and to maybe even cloak it and allow it to get into a host cell without being detected. There are some things called viroids, V-I-R-O-I-D. There are a lot of different types of viroids. Viroids are just a little piece of DNA or RNA, no protein whatsoever on them. Again, they're not living things. We do know of them causing plant disease in some cases, especially in citrus, but we don't know of any in animals at this point in time that cause disease. Doesn't mean they don't exist, but they're incredibly difficult to detect and work with, just like the prions, because they're not living, they're not reproducing, so they're hard to detect. It's definitely a needle in a haystack approach. So viral diseases, somewhere around 400 that are capable of infecting plants. There's a handful that are economically serious to us. The infection, again, does remain forever, even if the plant is not killed or dramatically affected in a negative way. We're not going to be able to cure this, just like trying to kill or cure viral infections in animals. We're not going to get rid of them. Viruses are transmitted from plant to plant by living things, by vectors, insects, mites, fungi even, and nematodes, which we're going to talk a little bit more about in a few minutes, can all transmit plant diseases that are viruses. Throw humans in the mix here too, because we tend to spread them around quite often. Rubbing, abrasion, pruning, grafting, any of these types of methods, whether on purpose or by accident, where we're rubbing and doing damage, whether we're seeing it or not, to plant tissues, means we've got a wound and a potential opening to spread viral disease. And on occasion, we do transmit them in infected seed as well. So just to give you an idea here, let's say we've got an aphid that aphid comes along, it's piercing and sucking, it pierces into an infected plant, and as it is taking a drink of those nutrients, it pulls up viral particles, goes on and sits over on a non-infected plant, pierces, and when it pierces, it spits just a little bit with that salivary gland, and when it spits back in, it spits in a viral particle, and there you got viral disease spread from one plant to another. Very, very simple, but you can see the path with that red arrow that viral pathogen might take, and its route in and out of a plant and in and out of the insect. So virus disease symptoms, there's 4 categories, and this is pretty much the progression of viral diseases. First, lack of chlorophyll. In normally green tissues, you get yellowing, you get chlorosis, like you see in that upper top picture up there. The second stage of viral infection is usually stunting or growth inhibition, and you can see the difference there between those 2 turnips. One's infected, one is not, same age of plants, treated exactly the same. Distortions, again, on the fruit, the little nodules that form, deformations. The size can be completely wrong. It can be twisted. Leaves, stems, all these types of things can be distorted and not functioning properly. Sometimes we like it and it stays around. Other times it causes us a big problem. But in the later stages of a serious viral disease, we get necrotic lesions or areas where the cells have died due to replication of the virus in those areas, and then you get a rotten area, kind of like what you saw back there on the cabbages earlier on. So nematodes, I will throw in just a very brief discussion of nematodes. Nematodes are microscopic, for the most part, roundworms. They are not leeches, they're not earthworms, they're in their own phylum of organisms. You can find some in the water. You find a lot of them in the soil. Some are decomposers and are very good organisms. Others can cause diseases in plants or malfunctions. So we tend to study them with the bacteria, the fungi, and the viruses. And they can certainly act as a vector for pathogens. Mostly clear to transparent, but they have the ability to pierce into a plant and do a lot of different types of damage in some ways. Some of them are able to infect roots, bulbs. Some are foliar feeding, not many, but there are a few. But as they feed, they weaken and they stress the plant and they predispose it to other problems. You can get root knots with root knot nematodes where the roots are just not capable of functioning properly. Sting nematodes where the nematode literally will inject toxins into the plant and control the development of the plant, leading in most cases to death. It gets the plant to do what it wants to do, not what you or the plant want it to do. But these are very frequently spread around by splashing water, infested soil, plant parks that are carrying the nematodes around as well. So you got root nematodes. You can see some of the damage up there where the peripheral roots have just been gnawed off. They've been killed and consumed in some cases, leading to nutrient and moisture disorders. Root lesion nematodes cause terrible malfunction. Some are burrowing and some are root knot or sting nematodes. So a variety of issues that we can have or disease-like symptoms caused by nematodes. So let's talk just a little bit about trying to control plant diseases. We have that environmental corner or piece point on the triangle. And some of these are easier to control than others, but a lot of these are the abiotic factors we talked about that humans can have some impact on, or areas where we can take advantage and maybe make the environment inhospitable to a pathogen. You've got the pathogen, you've got the host. If you can manipulate the environment and take that part of the equation out, you can limit disease. So we're talking moisture, temperature, us And vectors as being dispersal agents, soil pH, compaction, lots of different things that come into play here. Temperature certainly affects growth rates not only of the plants but also of the pathogens. And you typically have pathogens adapted to a particular range of temperatures. Anything hotter or colder than their optimal range, and they have trouble causing plant disease. We do know that temperature and humidity, when we tend to get very high temperatures and high humidity, especially in the overnight hours, it's a good indicator that we need to start scouting for disease, looking for symptoms very early on, and taking precautions trying to prevent the spread of disease. And that might be as simple as just removing a plant that is exhibiting some symptoms and or some signs so that it doesn't spread to others. Even refrigeration can be important for management of disease. When we talk post-harvest, say you've got strawberries, the quicker you can get them picked and into a refrigerator, you're going to slow down the growth rate of the inoculum of any pathogens that might be there to eat on them. And there are plenty of them. Dispersal agents. We know especially the bacteria and the viruses need natural openings or wounds. And if we can limit those wounds, we can limit dispersal. So we know wind, we know water, capable of spreading these types of things around, as well as insects and other animals can be vectors and infected plant parts. But people and machinery, our hands, our tools, our pruning equipment, even something like a cigarette could contain a viral particle. And if we are holding that cigarette and that tobacco had a particular plant virus in it, it's not going to hurt you whatsoever. But if that got onto our fingers and then we go rubbing on our plants or grafting or propagating in some way or pruning, we could spread that viral particle to an uninfected plant. If you were to work in tobacco fields, if anybody's ever worked in tobacco fields like we do or did at one point up in Kentucky, you'll know that one of the easiest ways to spread something like tobacco mosaic virus was through cigarettes. And so in a lot of cases where you have tobacco farms, you're not allowed to smoke and work in that tobacco field. So soil factors, lots of different factors. pH is one of the easiest and first things that you should think of when it comes to trying to prevent plant disease. You know that if pH is way too high or way too low for a particular plant species, that plant's not going to take up nutrients. Or it could even become toxic in some cases, depending on the pH. So that plant's not going to be in a very fun environment, conducive to growth, and it's going to then be susceptible to disease, which it's constantly exposed to. So it's kind of like, in a sense, building up that plant's immunity system so that it can withstand some of the disease pressure. Soil texture can come into play, whether you're talking clays, sands, loams. Compaction is a big issue, especially around new construction with home lawns. That compacted soil prevents the roots from moving around. So if the roots are not happy, the rest of the plant's not happy. Certainly soil moisture. And then if you've got a history of disease in a particular location or that inoculum, you're probably going to continually have problems there. That's why rotation of plants, and not even just the cultivar in a garden, but rotating the entire garden if you can from one spot to another, if you have enough space to do that, will help lower disease pressure as well. And again, not monocropping. Going in with genetically homogeneous crops, which can favor plant disease, is a very good way at preventing. So management of plant diseases, lots of strategies. You can till the soil, you can rotate your plants, rotate your garden, that's going to eliminate, reduce inoculum, that's preventative, that's very good. One of the best ways to give your plants the best start is one of the principles of Florida-friendly landscaping, putting the right plant in the right place, and again, here comes time in, at the right time. That's going to shorten its exposure to unfavorable conditions for the plant and favorable conditions for the pathogen. Controlling moisture and increasing airflow when possible. How many times have you seen a new planting in a landscape to where it is overcrowded? And over time, those plants are not going to be able to have airflow around them. And that's going to be a prime spot then for disease to take a serious toll. So not overplanting, or in some cases thinning out plants when possible, when necessary, will help to reduce disease pressure and controlling moisture. Easier said than done, but you can do this easily with an irrigation system out in your lawn or in greenhouses, or something just as simple as using the micros instead of overhead irrigation where you're leaving the leaf wet. You're increasing humidity and leaving a prime spot for fungal and bacterial pathogens. So a handful of very simple things that we can be doing that all lead to prevention of disease. Again, that's where we want to be headed, not trying to cure. Sanitation, very important. I do believe that in our area, a lot of the problems we see in landscape plants is often due to having pruning equipment that's not been sanitized. It should be sanitized from one plant to the next. Not one yard to the next, but one plant to the next to be effective. And you can use something just as simple as rubbing alcohol. I'm not a big fan of bleach because it's really hard on your tools. But diluted 10% bleach solution is effective in many cases of controlling pathogens. Or even using hand sanitizer, if necessary, can help disinfect our tools. Using fungicides is an option, again, not curatively, but preventively. Going with the lowest toxicity first if it warrants the use of fungicides, and fungicides are not going to control viruses or bacteria. There's a reason they're called fungicides. Going with host plants that we know are resistant to particularly common pathogens in our location. And you can find this information on tomato plants, on pepper plants, on squash plants that are being sold at your local retailer. They'll have little symbols on there that will tell you this is resistant or immune to something like verticillium wilt. Those are the ones you want to go with. Rotating crops, having good cultural practices in place, controlling weeds. This is a huge one. I always tell especially the agronomic producers, if they'll control the weeds in their crops, they will reduce their disease pressure by 85 to 90%. Weeds compete with plants for nutrients and moisture, weakening the plants. So they're competing with the desirable plants. They also harbor disease pathogens and insects that carry disease. Control the weeds, you'll control the disease very easily. Controlling temperature when it's possible, or at least planting based on seasonality. Going with biological controls, so using things like ladybugs and promoting their use in the garden, trying to reduce the use of pesticides, and those are necessary in many cases to get a situation under control, but trying to promote our biologicals is a very good way to help us in our gardens and our home landscapes keep a lot of diseases under control. And even going with something as simple as compost. Continually adding compost into our small little gardens. That's going to reduce nematode infections, which will also reduce a lot of disease pressure. And it keeps the plants healthier at the same time. Anything we can do to improve plant health and nutrition is going to lower the potential for disease. So again, with sanitation, we're trying to exclude the pests. Avoid sites we know that are infested. Use clean soil. Use good planting material, seeds, tools that are clean. Inspect, scout, quarantine. Remove infected plants and old plants, debris. Get those out of the way so that we're starting out with the best potential and cleanest of stock as we possibly can. And again, there's a wide variety of fungicides that are out there on the market And we do want to be careful with those and use them in a preventative, not curative type of methodology. Using resistant host plants that are resistant. Let's not get too much over into vertical and horizontal resistance. I don't think we need to go there. But this is just one piece of the puzzle in helping us take that host out of the triangle equation while still having our plants at the same time. Looking for hosts that are resistant. Rotating our crops. Getting rid of all those different residues that can be left behind and harboring pathogens. Very critical, very easy thing to do. And then again, trying to minimize the spread and the dispersal of these types of organisms through wind, through rain, with the use of drip versus overhead. The timing of our planting, maybe increasing the width of row spacing in our gardens or in our fields to improve airflow, and getting rid of alternative hosts for viruses and other types of diseases. And what I mean by alternatives are the weeds, for sure. If you can, best way to control temperature would be through soil solarization or sterilization techniques. It's easier for some than others. Usually more of an agronomic cropping technique than it would be for homeowners. But there are a lot of different solarization and sterilization techniques that are out there. And refrigeration. Slow down the disease progress in anything we've harvested and maybe even over in our seeds at the same time. So sanitization, the use of fungicides when necessary, rotating our crops, getting plants that are resistant when possible, using good sound cultural practices, and controlling temperature and moisture wherever possible, and promoting biologicals is going to absolutely improve plant health and nutrition. And taking time to sleuth in the garden will help us start to understand the normal plant, what it should look like. We should know what a normal squash plant looks like in comparison to one that might be diseased. Sometimes plants have very unusual parts and components that are very natural. And knowing what disease looks like and what it doesn't look like is very important. So remember, disease is not the norm. You need to know the normal plant. You need to first look for nutritional deficiencies. They're way more common than disease. Look for cultural explanations. Maybe the dog is loose and going to the facilities in the wrong place, and it's not a disease but a dog problem out in the yard. Look for signs. Look for symptoms, two different things, and can you associate the two? If you do need help, if you've got homeowners that need a more definite diagnosis, you can submit samples. There is a form online at UF. You can submit samples to the Plant Disease Diagnostic Clinics, $40 a sample. For turf grass, sometimes we need a very, very rapid diagnosis where we don't lose the whole lawn or turf, and that's $75 a sample. And you can also use DETAS to post pictures and look for answers from experts and extension agents around the state. So I thank you all very, very much for listening to me ramble on about disease in plants and how to prevent it, hopefully. If you've got questions, please feel free to reach out to me. My email address is wcelmore, ELMORE, @ufl.edu. I hope you enjoy the rest of your Master Gardener training, and I'm glad to welcome you to the family of volunteers. Thank you.